Here we go. This is chapter 3, and we're going to be discussing, uh, really kind of comparing uh, eukaryotes to uh, the prokaryotes, and we'll see how that goes. But there's one thing I wanted to bring up that should be on the top of everyone's mind as you have taken exams and the like. Let me let you in on a secret. This is backed on behind all sorts of data. And the idea of why I do reading assignments and various projects or worksheets is right here: memory. Now, you can look this up. You can Ebbinghaus is somebody you can Google. But the idea is the more you refresh your memory, the longer term that memory will be. And look at the time commitment: 10 minutes, 5 minutes, 2 to 4 minutes. Now you see why I assign these things to get your focused attention, and it's somewhat redundant because, well, that's how memory works, is redundancy. So if you want to, my goal is for you to walk out of the door, or if you were in the building, but if you come offline and you go on to your next task, you're going to remember micro, because I have tried to pull every trick I know to get your memory to be long term, and that's the deal. Exams help with that, and studying, writing, critical thinking, all those things help that memory, and then it becomes long term. And the more you do it, guess what? The easier it is, the shorter time you have to refresh that memory. So, just a tip from your Uncle Ed there. Uh, the neuroactive potential of the human gut microbiota. This is a paper that came out in February 2019. And what they did is, it was a large scale. This is why I wanted to look at it. And what this shows, if you take a regular regimen of various of a good probiotic, one that contains at least 5 or 6. I take one that has 16 different microorganisms, and what this does, it helps fight clinical depression, and there's a huge amount of data behind that. And if you're under stress, you see, what I'm finding is whatever shifts the population of the bacteria in your gut, usually it's away from a better state, unless you're taking a probiotic, because stressors, various different types of foods, antibiotics, medications, the list goes on and on and on, will change that gut flora, and it may have bacteria producing chemicals that are detrimental to your overall body system. So I wanted to bring this up. Of course, I always talk with these matters with your doctor, but it's just what I try to do is bring up ideas and concepts that you want to talk with your doctor about. And this is definitely one that probiotics are generally regarded as safe. That's under the FDA. It's GRAS, GRAS, and it's suspected to be safe for you to do this. So anyhow, still get a doctor's approval. So in the various papers that go on this, I have found more and more supportive. So it's just one of many different types of things I'm going to bring up during the course of this class that suggests how important our microbiota is in our gut. And so mood, all sorts of things. So anyhow, I'm going to try to start every class or discussion with some point of fact or some interest that if you want to write an extra credit report on the benefits of gut flora or probiotics, I certainly would be open to that. Give me a proposal and we'll go from there. So, bacteria and archaea, and this is chapter 3. Chapter 4 is on bacteria and eukaryotes, and sorry about that. I'm kind of excited about both chapters, actually. These are all important things, but as we go along, everything that we had done so far, especially chapter 11 We talked about chapter 1. Those are important for the reasons why we want to study more about the types of, uh, let's see, wow, the structures that bacteria possess. Uh, we're going to talk about the major shapes, and the shapes are important. Provide 4 terms that describe these arrangements. So we need to be able to talk about bacteria and how they're arranged. It's all about vocabulary. And so I usually, when I take notes, I use the Cornell method of note taking. And if you're not familiar with that, I would suggest Googling Cornell note taking. It'll take you to a site. It shows you how to draw on a piece of paper certain segments. One of those regions or areas of those segments that you draw on that page is terminology. And that's so important. The more you write it, the more you remember it. Memory. Okay, so bacteria and archaea are different. Now, I'm going to tell you up front, I'm not as concerned that you know about archaea because they're not clinically important. The temperatures at which bodies function in humans and healthcare, it makes archaea really irrelevant for us. It's nice to know about it. It's a great exercise, and it's part of our objectives. And so I'm going to take it to the bare bones of what you need to know on that. But we're going to build up more information about what we know about bacteria. That is what I want you to spend time on, and that's what's more important for us because they are relevant for clinical scenarios. So DNA is packaged. The lack of nucleus is different. The makeup of their cell wall, peptidoglycan, are unique chemicals and internal structures. And both of the archaea and bacteria sort of are this different from eukaryotes. So that makes it easy because the next chapter we're going to be talking about eukaryotes and how we differ. Now, well, I don't want to jump ahead. Let's go ahead and just go on. Bacterial cells possess cytoplasmic membrane. Cytocell, plasmic is sort of a containment, a plastic container if you want to think of it that way. And ick is a person, place, or thing, and it's a membrane. So it's a membrane that sort of holds in this cytoplasm, or the gooey, chewy center of the bacteria. Cytoplasm is that material. Plasm is a really interesting word. Greek words have a very nebulous meaning. When I studied Greek, I took it after I started studying Latin. Now, Latin's very precise. And you look at the word, you conjugate the word, and you know where you are. In Greek, it's more of a feeling. It's sort of like Japanese in a way, that if you look at the way the characters or Chinese characters are written, it conveys a story, and that's sort of how the Greek language is a little bit. So plasma just makes up this sort of mysterious stuff, and so it's stuff that make up cells. Ribosomes, we all know what ribosomes are, and those, of course, are the manufacturing components of a cell. Cytoskeleton is keeping the organism from getting squished. The skeleton is the sort of the X, Y, and Z strength of a cell, so stacking and pressure and those sorts of things won't flatten them. And so we've got to have that. And or a few chromosomes. Now, what they mean by that? Well, there are some bacteria that we may get into the lab about when we do some UV resistance. There are microorganisms that have multiple copies of the chromosome, and it makes them very hardy. So you can mutate the chromosome, but you got to mutate all 4 of them at the same place. Probably not going to happen. So they tend to be a little bit more hardy out there, and that's sort of the reasons they do. But for the most part, other chromosomes are plasmids, which are smaller circular pieces of DNA that confer antibiotic resistance or adherence mechanisms or anything else. They're sort of like they love rummage sales or these sales bins. They'll try on. They're not proud. They're not. They'll drop their drawers and put on somebody else's drawers right there. It won't They'll change genes all the time. So they're not bashful. Bacteria will do whatever it takes to survive, and they're very lean, mean fighting machines. So uh, most bacteria possess cell walls of some kind. Now, when we talk about cell wall, we're talking about a chemical composition that gives it some fortitude. A surface coating called a glycocalyx, sugar, and calyx is sort of like I don't know. You think of a calyx as maybe something that holds the glucose structures, and it's a little bit tougher. And we're going to talk more about it. Not some, but not all bacteria possess flagella, pili, and fimbriae. Now, all of these are external structures that do certain things. There is a little bit of confusion between pili and fimbriae. Pili are usually referred to as mechanisms for adherence, but also they've been used classically as how to transmit in one of the modes of DNA transfer is conjugation, and it goes through this sort of tube. And fimbriae are usually referring to adherence mechanisms, so little fimbriae or hairs. The pili are much longer and thicker, and fimbriae are hairy, sort of like that the baby hair on the face. Children that are just born new, you see these little dinky hairs. There's a name for that, I forget, but you probably remember. But flagellas are used for, they whip them and they spin them, and they use it for transportation. So it's their little Evinrude motor that they all have, and how many motors and things, there's names for all that, of course, and we'll talk about it. An outer membrane, nanowires and nanotubes. There's actually documentation that bacteria communicate through, it's sort of like a string and two cups, and there's a vibration that occurs. I'm not kidding. So there are some forms of communications that certain bacteria will use, and in addition to other things that these nanotubes and nanowires, plasmids, and those are the smaller pieces of DNA, usually circular always. They may have come from viruses, bacteriophages, and they form a circle when they get in, and that's a plasmid. Usually confer antibiotic resistance, but it can contain or reserve for other features. Inclusions are just sort of like little storage bins. It's like getting a bin to store something, inclusions for various things. Endospores, these are the preppers, the bacteria preppers. In other words, they'll go into this storage as a spore, and they can withstand a long period of time. And we've talked about it, but endospores are a problem, and we've actually kind of based our autoclaving or making our sterility standards based on spores and killing the spores, and that's sort of one of the toughest things we know of. There's more. Microcomponents, compartments, these are small little compartments. They're not really sort of membrane bound, but they're kind of held together by sort of just a very thin sheet of something. It could be a fat, or it could be something. Anyhow, we'll come back to that. So I'll give you a few seconds to memorize this, but actually it's a good summary of various things, and I've added that as an image on the instructor page there, and you can see it's important. We talk about S layers. I like to think of the S layer as sort of the seal. I don't know, it's like a hermetic seal. Fimbriae, those are used for attachment, hair-like structures. The outer membrane, it's an extra membrane similar to a cytoplasmic membrane, but also containing lipopolysaccharides. And so you look where that is. So we have this seal, the S layer, and then under that we have this outer membrane. And then we go into the cell wall, which is a semi-grid casing, I guess is what they use, but it's more of just it's a chemical structure that provides structural support and shape for the cell. So it sort of provides that. And round things tend to be naturally stronger than flat or other types of structures. So nature seems to always form sort of like a balloon shape. Now this is a elongated balloon, but anyhow, it's circular. You get the idea. The cytoskeleton are long fibers of proteins that encircle, and these give it that compression resistance ability. The cytoskeleton. There's the pilus. Compare the pilus to the fimbriae up here, and you can see it's a tube. It's a little bit thicker, and well, it's usually a little bit longer than the fimbriae. It's however someone the artist depicts it, but it can transfer a double-stranded piece of DNA through it. Now, not circular, but it goes linear, and then on the other side it forms back to circular. The glycocalyx is this coating of layer of molecules external to the cell wall. It serves as a protective adhesive or receptor. Functions, it be like a fit tightly like a capsule or be very loose like a slime layer. Depends on the environment. There's an inclusion of granulous stored nutrients, some kind of fat or phospholipid or phosphate or something. And then there's a plasmid, a circular piece of DNA, and bacterial microcomponent compartments. It's sort of like a you're on your dashboard or something, having a little something. These micro compartments used to localize some enzymes, and they kind of keep them together. You know, it's here or there. It's whatever. There's a flagella. So look at the flagella as compared to the pilus. Um, the pili are more than one. Pilus is one. There's the flagella, and the flagella spins. And when it does, I'm going to explain what's going on. If it spins in one direction, it sort of propels it forward. If it spins it in the opposite direction, it flails. And when it does, it changes the direction at which the bacteria will move. These bacteria are pretty smart. Nanotubes, nanowires, they say membrane extensions that allow bacteria to transmit electrons or nutrients or information to another environment. So endospore is not shown, but it's a dormant body. If something has caused the organism to say, "Hey, look, we got problems," these organisms will hunker down and not have problems. So, glycocalyx is a glucose and sort of a matrix material that's resistant. You have the S layer, which is sort of like the seal peptidoglycan cell wall, then the cytoplasmic membrane, which is yet another membrane that's permeable, semi-permeable, and there's the chewy, gooey inside with the cytosol. That you can see things are kind of just there. No compartments, you know, like a nucleus or Golgi apparatus. We don't have any of that. And it's sort of like a bathtub where everything's floating and kind of just there. And anyhow, it makes it sound kind of, well, that's what it is. Most bacteria function as independent single-celled unicellular organisms. They act as a group in colonies or biofilms. That's a really important word. I would highlight that, underline it, draw arrows to it. A biofilm is important. These are a culmination. You can write next to the biofilms, you can write synergistic, because that's what it is. You have multiple organisms synergistically acting together in concert to do something. Some communicate through nanotubes, which there's chemicals, communication. We're still trying to understand what's going on there. Bacteria have an average size of about 1 micron. Rods have a length of about 2 microns, a width of 1. Pleomorphism variation in the size and shape of cells of a singular species due to nutritional genetic differences. Pleomorphism is something we need to think of. We've seen pleo-colored cats with a different coloring variations of various types. A pleomorphic bacteria can change how they appear based on the nutrients in the environment. And it's frustrating for a microbiologist sometimes, but we have to understand that when we take them from one environment and put them in another, we expect them to behave the same, and that's not the case. They'll have different strategies now for which enzymes are expressed and things to the new environment, and as a result, the way they look might change. They don't really care how they look. It's just sort of like me with my hair going wild because I don't really care. I need to brush it, but bacteria just you get to the point where uh, deal with it, that sort of thing. So here are the shapes. Cocci, a round, not perfectly round in all cases. Can be look like little kernels of corn, things like that. Rods or bacillus, again, they could be a little bit odd shaped. There's gets too far, we actually can call that shape something different. But for right now, bacillus is a rod shape. Coccibacillus is sort of a round rod. Now, this kind of could be considered a coccibacillus. Vibrios, they curve and are called vibrio. They look like commas under a microscope. Spirillum is a bacterium having slightly curled or a spiral-shaped body, a spirillum, a corkscrew-like thing. Campylobacter jejuni is a great example of a spirillum. A spirochete, it contains a periplasmic flagella. Now this is really, really weird, and I'm going to explain it now, but I'll go over it many times. A spirochete. Think of the outside of the microorganism spinning, but the inside stays still. It's not turning. And think of it as sort of like a motorized auger that's drilling right into the ground. An auger kind of drills a corkscrew into the ground. That's kind of what's going on here. So a spirochete. Borrelia burgdorferi, the causative agent for Lyme disease, is a spirochete. Treponema pallidum causes syphilis, is a spirochete. Part of the pathogenesis is it literally does sort of screw itself right into your tissues, and that's how it does it. Branching filaments. So some bacteria, not very many, like streptomycetes and things, they are bacteria, but boy, they'll fool a newbie type of clinical microbiologist because they say, oh, this is fungi. And you got to be careful, and you don't see any filament segments here, and that's your first clue. You might see the fruiting bodies, but you don't see the segmentation, which is characteristic of fungi. So if they branch and they have these little roundings at the end, you might want to be thinking and slowing down and run some tests that it's a streptomycetes. There's always an exception to everything, right? So the arrangement of the cocci, or the round ones, are single. We can have diplococcus, which really echo on how they divide. And when they're in a hurry, when they're dividing every 20 minutes, sometimes they finish without really finishing and move on to the next. And sometimes it leaves certain bacteria kind of sticking together a little bit, and it seems to be a characteristic or a modus operandi, and it's a diplococcus. So you always see it in pairs or tetrads, same thing. Staphylococcus or micrococcus, irregular clusters. Staph always kind of looks like a grape-like clustering of cocci. It looks like a thing of grapes. Micrococci, not so much. They're more irregular groupings. But staph will be more like a cluster that looks like grapes. Streptococcus are in chains. Strepto, think of it as a chain of cocci. Sarcinia is a cubical pack of 8, 16 or more. It's in that sort of cluster of those Sarcinia. So this is a weird, it's a Greek word, and sarc, and I think there's a Latin version of the same thing. And I think that's where the word sardines comes from, but anyhow, I'm not sure about that. I have to check that. But here's what I meant by the division plane. So if you have them dividing in one plane, so this one's going that way and that one's going that way, you get two. And if it continues, you see the strep. If the division's in two different perpendiculars, you might see tetrads forming as a result, or Sarcinia package of these, and it's the way they kind of stick. Division in several planes, you get your grape-like clusters, or your kind of grouping like the micrococci do that. So it's all about how it goes through division and its characteristic genetically. The arrangements of bacilli, which is a little bit more simple, I guess. Diplobacilli are pairs, which ends are attached, and so you see some that do that. Streptobacillus are chains of several, and we see Streptobacillus pretty often, I guess. Palisades are cells of chains that remain partially attached by a small hinge region, like you see here, and those are palisades, and they're just weird looking, and it's all about how they divide and that sort of thing. So I included a picture in the discussion for your instructor, and this kind of goes over all that I've talked about in a nice, very concise looking chart. And so I thought that might be useful and gives you an example of each. Here's the spirochete that I was talking about and how they auger or drill themselves into the skin or tissue. That's pretty nasty. I've looked at Treponema pallidum under microscopes. They are highly motile. They know how to zip around. They're fast. So we're going to look at the structure and function of 6 different structures found in the exterior of bacteria cell. And what I decided to do, because I blabbed on to the point where you're probably ready or yawning already, and some of you have already dozed off. I have a doze off meter attached to my computer here, and I can see that some of you are. But I'm going to stop, and I will pick up on section 3.2. Now, I've rearranged how I post these. It'll all be in the one area so that you can look for the videos, because I sometimes do a video on a section, and sometimes the section's so short that that's kind of not worthy of your time, so I kind of include them together. So that's why I just kind of separated those out, so you'll see where the videos and various things might be for you to the slides and things all in one convenient area, and so you can go from there. So that's where we are for now, and I will pick up on section 3.2 in my next video. So study hard. Remember, just 10 minutes and you refresh that memory, and you're building towards long-term memory, and it's amazing what happens during an exam time. So I'll leave it at that.